welcome everybody. My name's Darrell Bullock, and this is session 3 of the NBC C. Brown Bagger series. And we've got a couple of excellent speakers today for you, so we'll jump right into the program. Let's see. As a reminder, we still have the website up and running. We don't do a whole lot with the website any longer, but we still do put all of the brown bagger recordings and all the archives are still on that site, as well as the sire selection manual. I told the group last week, and I'll let you guys know again, we have printed the 3rd edition of the sire selection manual, and we do make that available to any educators that would like a box of them. I can ship you a box for free, thanks to the Beef Improvement Federation funds that for us. So if you would like a box of the manuals, just shoot me an email at dbullock@uky.edu, and I'd be happy to get a box out to you. As I said, I'm your host, and our first speaker today is Juan Pedro Steibel, and I'm going to read you Juan's little bio here because I thought it was very interesting, and so I usually pick and choose, but I'm going to embarrass him a little bit, and I'm going to read what we have here. Juan got his BS in agronomy from the University of La Pampa, I think, in Argentina, and his master's in biometry from University of Buenos Aires in Argentina, and his PhD in animal science from Michigan State. His main research and teaching interests are in the area of quantitative genetics, genomics, and phenomics in animal populations. Specifically, Juan's research involves the development and adaptation of statistical and computational methods for joint analysis of multiomics, including transcriptomic, phenomic, and genomic data with ultimate goal of improving global food security. Most of his applied work has been in the swine production systems, but he has also worked and published on other animal production systems and on wildlife and fisheries species. His program is funded by commodity groups, breeder associations, and NIFA. Juan is the J. Lush Endowed Chair of Animal Breeding and Genetics at Iowa State University, where he is a professor in the Department of Animal Sciences. Before that, he was a professor of animal science and fisheries and wildlife at Michigan State University. And Juan, you were telling me that you've been in your new position at Iowa State for about 2 months now. So welcome to Iowa State and welcome to our program. We're pleased that you're with us today to share, like I mentioned to you earlier, I think a little bit deeper topic than a lot of times we get into, but I'm very interested to hear what you have for us today. So I'll turn it over to you.
Thank you very much, Doug, and thank you everyone in this consortium, and especially Matt Spangler, who invited me to give this presentation today. I hope to fulfill your expectations, whatever those are. I will say that even though I worked a lot on big production systems, at heart, I love big production because I am originally from Argentina. And to me, if you ask me about animal science, I thought it was only beef cattle before I came to the US. But circumstances sometimes take you on a path that might be different from what you came from. So I was asked by Matt to talk about what you see in this slide, an introduction to machine learning and other artificial intelligence with potential application to beef cattle. So let's go into it. So first of all, what is artificial intelligence? This is a very broad definition, what you see there. And artificial intelligence is any technique that enables machines, computers, most 99% of the cases, but they could be other machines, machines to mimic human intelligence. So this means to mimic the way in which we make decisions, which means that this can be from very simple decision rule, like if you see this, do that, if you see this, do that, based on some human knowledge that is put into the machine, or it could be pretty sophisticated decision rule based on looking at data, and doing something with the data, the information that is coming from the data, and then making decisions. So that's what artificial intelligence is. It's a pretty vague definition. A subset of artificial intelligence is machine learning. That's what I try to mean here with these like kind of Venn diagrams nested within each other. So machine learning is a special or a subset of artificial intelligence, a special case where statistical techniques, models, or even computational algorithms are used so that the machines running these algorithms can improve the performance of the assigned task as they look at more and more and more data. And I will try to build on the knowledge that I assume most of you will have. An example of that would be very simple, least square regression. Least square linear regression is a statistical technique that you provide data, there is input data in the form of predictors and response variables. And then there is a numerical / statistical algorithm inside that will do something with the data and come up with model parameters, which is the parameters to define, for example, a straight line. And then that is used to predict something. So that could be a simple prediction device that is machine learning. As you give more and more and more data, the machine will learn more about your system in the form of a slope, summarized with a slope and an intercept. Pretty simple, but effective. There are some that are very sophisticated. In particular, a subset of machine learning, or a particular case of machine learning, is called deep learning. Now we are hearing a lot about deep learning. Deep learning is a type of machine learning where deep neural networks are used for learning. And I will stop here. I know that someone will say, what are you talking about, deep neural networks? We'll see this in the next few slides. But when someone talks about deep learning, they are talking about the particular case of machine learning, which is one of the ways in which artificial intelligence could be implemented. It's a pretty small set within the big set of artificial intelligence. But we'll talk about that quite a bit today, because I think it's novel and cool for us, for animal readers and animal scientists. So before we go into this, let's review what are the most common statistical tasks that we do with our models, with our numerical and statistical models. In general, we have two types of goals. Can you see my pointer in the slides? Can you see my arrow?
Yes.
Okay, thank you. So one of the most common tasks is inference. And in inference, we focus on non-observable, on model parameters, and we want to learn something about the system. I will talk more about this next. But the other statistical task, is prediction. In prediction, we focus on future observations. Right? So these are two different tasks. One is to do inferences about model parameters, the other one is to do prediction about observables. In animal breeding and genetics, in particular, and also in other areas of animal science, for example, precision livestock farming, there is a heavy emphasis in predictions. We are almost all about predictions. We want to predict stuff. We want to predict observables. But we sometimes want to infer things. I will talk about this in a second. On the other side, on management, reproduction management, nutrition, behavior management, we are mostly focused about inferences. We want to learn about the system and in the model of system parameters. So most of my talk today will be about prediction, but I think it's worth going over the differences. One example of prediction that we use a lot in animal breeding and genetics is predicting breeding values. And also we might try to predict phenotypes if we want to use this prediction for management in addition to selection. The other example for inference for us for application, I have here finding candidate variants and genes and pathways that underlie the actual phenotypic variation. So the genetic causes of phenotypic variation, that would be inference. Another type of inference that is very common for us is estimating the heritability of the trait or the genetic correlation of the trait. We want to estimate a non-observable, a model parameter, because that has an interpretation in terms of our theory, of the models that we use to base our theory. But our goal today will be mostly about predicting new outcomes. And so when we want to predict new outcomes using whatever type of statistical or machine learning model that we can think of, the goal is prediction accuracy. We want to maximize prediction accuracy. So we want the predicted values to be as close as possible to the true value of what we want to predict. So the focus and the emphasis is on observable, on future data. The EPDs of a bull are actually predictions of the performance of the progeny of that bull. So we are trying to predict future events, right? It's predicted on the bull, but it's based on what will happen after that animal is used to sire progeny. And then when we have models, we still use models, statistical models or other type of machine learning models. And we want to check the models to see how valid our models are. The way in which we do that is through cross-validation in general. And sometimes we might have uncertainty about the model. We are not sure which model to use. We might have several models. The way that we deal with that in general is averaging out across models. So we might base the prediction on a single model, but in many times embedded in our modeling, there is some sort of averaging of different parameter spaces. So model averaging is a good property to look for in our devices, predictive devices. And for prediction, the best performance, the best type of data that we can have is very large. These are data hungry, very, very large, extensive, and variable observational data. The contrary of that would be to base our predictive models in experimental designs. And that would be the case, for example, of a progeny test. If you progeny test bulls, you are doing prediction, but based on an experimental design. It's great, it works great, but sometimes the amount of resources that you have to allocate to that is huge. And so we learn to do prediction using just observational data. On the other side, if you want to do inference, it's much better to have a well-designed experiment. But that's for another talk, another day. Oops. All right. So if we focus on prediction, we need to focus on model accuracy. That is our end goal. We want to have accurate predictions, and for that, there's two ways to do it: better data or better modeling. And the better data, in the case of genomic prediction, for genomic data, it could be by increasing marker density. It could be having better annotations. If we use that in the models, yes, I still want to see that, but it could be a way. More individuals, more animals in our data set, multi-population, so to have as much diversity as we can, multi-breed predictions. I think beef breeders are championing some of that. Multi-breed prediction sometimes is the way to get more accurate. Sometimes it's single breed prediction, but with many, many more herds and so on. And sometimes we may want to incorporate some environmental data together with the genomics. Let's say we can exploit GID. For the phenomics, on the other side, one way to increase the quality of our data is to include more genotypes, novel genotypes that by genetic correlation can help with the prediction of other genotypes, more animals phenotyped. So automatic phenotyping becomes very important because we can measure more animals in more conditions. Some people also claim multi-layer omics, such as transcriptomics, etc. It was mentioned in the nice intro that it was made for my presentation that I work on that. Still, for the practical uses, I don't know how much we will get out of it, but it's something to consider, multi-layer omics. For the alternative modeling, this is what we do most of the time. We have the data is fixed, and we want to do the best model possible with the data that we have. So let's talk about alternative modeling. Let's talk about genomic prediction, right? Genomic prediction, this is a very generic genomic prediction model. We have our response here, response phenotypes. We have some fixed effects that represent population level averages, herd, year, season, or whatever you want to put in there that are easily to sort out. through metadata or data that we have available. And then we have this genetic additive component that is just a simple multiple regression component where we have marker data. This represents a genotype matrix, animals in the rows, markers in the columns, genotype data. And then we have the effect of each, the predicted effect of each marker on the phenotype of interest. And then, because we're interested in predicting the EPDs or breeding values, then what we do once we solve these equations, we just sum for a particular animal with a set of markers. We just multiply the marker effect by the marker status in an animal, and we sum over all the markers, and we get a breeding value. It's a very simple technique. Now, things get complicated when you have very large matrices, for example, with many more markers than animals, and then you cannot just estimate every marker effect. So that's where we have to start to put some restrictions into this model. And in the machine learning language, that is done in two ways. One is effect penalization or regularization is a type of model averaging. So we are averaging over possible values of those marker effects in different contexts. And or we can do marker selection. We can select just a few markers to do the prediction. And that's what some algorithms such as base B or base C or base C pi do. They just try to zero out some of these marker effects and leave the other ones with a value so that they are doing model selection. So this is just a generic predictive device. All right. Now, this genetic predictive device that I mentioned before can take several different forms depending on the assumption that we make for some of these components that I'm pointing at. One classical assumption is to assume that all the marker effects come from a common distribution. So they all have they are all randomly sampled from a common distribution of independent effects with independent, that means this identity matrix here, with a certain variance. And when we have that was called, at some point it was called GBLUP, genomic BLUP. It was early on in the development of genomic predictions, pointed out by colleagues from several institutions, including the USDA, ARS, and Iowa State, that this model is equivalent of a model where we model the animal effect, the random effect of the animal, the genetic additive effect, with a normal distribution, but with a variance covariance matrix now that is not an identity, but it's G. And that G is built based on multiplying the marker matrix by its transpose. And for these two to be equivalent, then we have to do some scaling and centering of the columns of the marker matrix. But these two models are equivalent. So this is an animal-centric GBLUP model, and this is a marker-centric GBLUP model. And if you do things in a certain way, you get the exact same prediction for the breeding values using either model. And this formulation is very appealing to many of us who come from the pre-genomic era, we've been training the pre-genomics, because this takes a very similar form to our traditional animal model, where our additive effects, genetic additive effects for the animals follow a normal distribution, but the variance covariance matrix is A, which is the numerator relationship matrix that is obtained conditional on the pedigree. And of course, now in this new era, the group from Georgia has brilliantly proposed a way to combine these two into something that they call H matrix. This is what we work with. Now, this GBLUP model, in the animal-centric way especially, is the single most used model for genomic prediction in commercial settings all over the world. I'm not talking about counting papers, I'm talking about counting actual breed evaluations. Probably an exception could be when there's more competition using non-GBLUP models. Probably could be in some breeding evaluation schemes where marker-centric models are more used. With some marker-centric models with different weighting of the markers are more used. But that's another story. By far, this is the most used one across production systems and in the animal-centric way, but presenting it in the marker-centric model, it's interesting to note some limitations of this model. One of the limitations is that this is just a linear model. So as you go from 0 doses of the favorable probability to 1 to 2, you expect a linear response. And maybe there could be a nonlinear response, right? Especially if there is dominance or something like that. Now, dominance could be added to this model, and even epistasis, and some people are working on this, probably in this group. But in general, in the bare bones way, it's a very simple model that doesn't account for that. Also, the SNP effects are assumed independent, and although there's been some work trying to use some local dependency models, etc., that's not really caught on. So it is a simple model, yet it works, right? So in the more recent years, it's been proposed that maybe we can use deep learning for that. And so I want to share some of the work that we did. And this work was done by my former student, Juan, at Michigan State. Now he works for Bayer. Unfortunately, he became a lettuce breeder, I think, or plant breeder produce area. But anyways, unfortunately, because we lost the brilliant animal breeder. But anyways, so the idea is that he wanted to work on this, on using deep learning models for doing things. And I told him, well, if you want to use deep learning model, use it for what I know what to do, which is genomic prediction. So I challenged him to do deep learning for genomic prediction. And this is an example of a deep neural network. The depth in this case is 2, so it has 2 layers. Basically, the input layer are the SNPs. So these are the 0, 1, 2s of the SNP, or -1, 0, 1 kind of scale. And then you see the output from each of these SNPs goes into a node. And then the output from those nodes goes into the next layer of nodes, and so on and so forth. I don't know if you can see it here, but each of these nodes will compute the output by taking a sum of the inputs of all these previous nodes, multiplying them by a coefficient, and then summing them up and transforming them with a nonlinear function. So basically, it's like a little teeny tiny regression within each one of this, and then a nonlinear transformation. And that is fed to the next one, and to the next one, and to the next one. At the end of the day, we get a prediction, and that prediction is compared with the observed value, and the prediction is compared with the observed value, and there is a loss function, which computes kind of a distance between the predicted and the observed one, and based on that, it will say we are doing well or not. If we are not doing well, we have to adjust the parameters, and the parameters are all these teeny tiny weights that you are seeing there. Another version is something called a convolutional neural network that works kind of similarly, but the transformations are a little bit different. They are called convolutions, and these are used for highly correlated data sets. And these are used a lot for image analysis. Okay. The problem is, because we want to adjust all these weights, the model parameters, there's a lot of model parameters, and they are very hard to find the optimal combination of them, because if you look at the loss function, there is a lot of local maxima and minima. And we are very familiar with it. We animal breeders are very familiar with this when we estimate variant components. So we know it's a hard problem. So one problem is to find this, and there's an algorithm for that is called back propagation and stochastic gradient descent. It's a couple of algorithms. I won't go into the details. So this is numerically, it's a very hard problem to solve. On top of that, we have to define a lot of things. We have to say how many layers, how many nodes in each layer, which type of nonlinear function. There is a lot of hyperparameters to define in one of these models. So for example, here is a list of hyperparameters. Some of them are structural properties of the network. Some of them are optimizer properties, how we find the linear parameters, and some of them are other penalties of the model. So working with Cedric Gondro, former colleague at Michigan State, and with Junjie, we applied something called the genetic algorithm to try to find the best combination of these parameters. What we found is that there is no best combination. These models are so over-parameterized that there is a bunch of models, 5, 10, 15 models that can do as well as any other of the 15 models. So depending on the data set, we found very different models. But in general, what we found is real data set require more complex models, more layers and more nodes than simulated data, for whatever that's worth. So let me go to this quickly. what we did find that if we optimize our model, these are our optimized model performance, we did much better than if we use an off-the-shelf model. So at that point, when you use deep learning, you have to spend a lot of time optimizing the model because there are no good models that are good across all the population. For example, these are models from other publications, and they didn't do that as well. However, when we try to compare everything to GBLAB, and GBLAB is the red bar here, any deep learning model at best tied with GBLAB. So you tell me why you do this if you will tie GBLAB. Well, that's something to look further into, but computer scientists say we are not trying hard enough. And I'm saying GBLAB works pretty well anyways. So I think there's more work coming. But deep learning model shines in some image analysis. So the question is, why it works so well for image analysis and it doesn't work so well for genomic prediction? And I always made the joke, there is not GBLAB for genomic prediction, for image analysis or computer vision. But anyways, the difference between us trying to apply deep learning into genomic prediction versus them, computer scientists applying deep learning for computer vision, is that they have this humongous reference data set. There is a data set called COCO, which is a data set for finding objects in images. It's a reference data. They have millions and millions of annotated images. Annotated images like this, where they have, you see here, the sheep and the dog, and they are all labeled and annotated. And then based on that, they have reference architectures that they use over and over again. The same happens for image classification algorithm, something called ImageNet dataset. It has 14 million images. So, and there are probably 10 different architectures for image classification that work very well, that were tested. So there is this bunch of already tested models and reference data that can be used as a foundation for building deep learning models in computer vision. We don't have that in genomic prediction. We don't have such a reference data set. So I will skip these slides because I'm close to time. But one thing I want to say is this. We don't have anything equivalent to these deep learning models, deep learning data sets and reference modeling in genomic prediction. We don't have that. Do we need this? Well, if we want to keep researching deep learning for genomic prediction, yes. If not, we abandon this and we keep working with our traditional statistical models. That's fine. But what is very unlikely is that our deep learning models will do any good, but if we try it still in a small data sets. We need to learn a way to represent the genome using deep learning machines better. One way could be, for example, taking something like the 1000 volts project and using an autoencoder that will find a reduced latent space using deep learning, a reduced representation of the genome, and then using that for prediction. That would be one way to do it, and we could talk about that in the future. This would be also useful for imputation, for example, although we have pretty good imputation methods. So I will skip the autoencoders, but my conclusion is that we do have ample experience with prediction problems, and we are used to use black box models such as the Bayesian alphabet. We call them black box. However, these deep learning models are much more cryptic and much more harder to interpret than any of the models that we've seen before. And so, even though machine learning has been proven to be very useful to improve accuracy of prediction in many applications, it looks like for our applications, if we want to use it, we would need much better data sets or larger data sets and a concerted effort in trying to create what they call a model zoo and I call pre-trained models so that we can make the most out of the data that we have. And if we want to apply deep learning for phenomics, that's the topic of another talk. We still would need to create our own reference data sets, but we have a lot to begin with because these data sets for general computer vision prediction, which we don't have for genomic prediction. So this is just my very broad overview of this machine learning, deep learning, and how it's working or not for doing what we know how to do, which is predicting breeding values and APVs.
Excellent. So if we have any questions, you guys know the procedure. You need to type them in the question and answer section if you would, and we'll get to those questions. So Juan, I guess, so in industries like the poultry industry, the pig industry and all, where they have really a better structure, I guess, in general, and lots more data. Are there being some successes there? I mean, do we have any hope in the beef industry where we're so segmented and I guess a little bit looser structure?
So a lot of people are trying these type of models for computer vision and for that, and there are quite a few stories of success, but I can also show you some where it doesn't work that well. So it's still in its infancy, but there is more work being done on that way for the genomic side. For the genomic prediction in poultry and swine breeding, we don't know what's been done. A lot of the work is done behind closed doors in corporate settings, in R&D in corporate settings. So we don't know. What I do think there is an opportunity for the beef industry is because the beef industry has its structure, is segmented, but it's a lot of breeding entities, and there is at least more national evaluations and more multi-breed evaluations in the beef than in the other sectors like poultry and in swine. I think there is a hope for that if a consortia wants to put together a data set, and there is a Thousand Bulls project and things like that. So I think there is more data and more potential to get better data from the beef industry than from the swine industry.
Let me see, we have something in okay, here we have something in the chat. If our traditional linear models work well for genomic prediction, should we invest time in advanced ML for machine learning, I guess, for prediction, or should we develop machine learning for the things our models do not work well for now? Phenotype prediction, phenotype collection data.
Yeah,
What's that?
Yeah, I completely agree with Matt. There is a question, but there is a suggestion there. There's an idea floating there, right? Yeah, I agree. And that's why we shifted our interest from these completely to, or our intentions of using these models from these completely to more computer vision and phenotyping and those sort of things. And I will save that for the invited talk from Matt that Matt gave to me for the summer. But yeah, I think if we don't have to do this, I think we have linear models that are kind of working. If there is a concern about modeling epistasis or things like that, we could go this route to see if epistasis could be modeled this way, or we could go with explicit modeling in a linear model. We can also do that. But then the real promise of this is for the phenotyping, is for image classification, for video segmentation, and for those things. I will talk about that in another occasion.
All right, we've got one more we'll get to, and then if you have others, you can type them in, but we're going to move on after this one. Will Schaefer asks, so far, most of what has been mentioned is genomic prediction. Is there much research on phenotype extraction? I imagine classification and count-based phenotypes are the low-hanging fruit. However, how about extracting quantitative traits from sensor data to automate phenotype collection? And then he wrote a clarification. I meant continuous phenotypes and not categorical.
For extracting continuous phenotypes, for instance, for extracting for extracting discrete phenotypes such as countings or just classifying images for behavioral phenotypes, and this animal is eating or is resting. For that, these deep learning models work very well. There is a lot of work published on that. For more continuous phenotypes, it's harder. It's harder to make these models work. And so a lot of the machine learning that is used for that is different types of machine learning, which are essentially like non-linear regression machines. And yes, so I think you can still try with the deep learning models, but deep learning models have a hard time in general predicting continuous variables with small data sets. And usually we have very small data sets, not in terms of number of observations, but in terms of the variability and the number of different cases that we have. It's hard to get all the different cases for a continuous variable. And that's why it's so hard for these models to predict that.
All right. Thank you very much, Juan. And I hope you'll stick around. And if somebody has another question, they can put it in and we'll take care of it at the end. But we need to move on. Our next talk today is the use of in-pen partial body weights in beef cattle by Dr. Mike MacNeil. And I know that a lot of you guys are familiar with Mike. I'm not going to, Mike, go in the interest of time, not going to go through your whole resume, but Mike had a long time career with USDA. I guess started at Mark and then out at Fort Keogh, and it has done a tremendous amount of work in those. He's been both the Continuing Service and Pioneer Award for the BIF, and he was also won the Animal Breeding Award from the American Society of Animal Sciences. All great honors, and Mike been following your work for quite some time now, and we really appreciate you. The latest that you've had to come out was this paper, I think, on your in-pen partial weight body weights in beef cattle, and we'd like for you to kind of update us. I imagine most of people have read the paper by now, but would like to hear your comments on that if you would, please. And I think you have control.
I believe I do, Derek. Thank you. So we're going to, let's see, doesn't like that. Dare, you took control. It says I have control. There. So the paper Dare mentioned is the reference is given here. I was asked by Vitelli to take a look at their in-pen body weight algorithms and do a quasi-independent evaluation of that. They were interested in having an international team assembled to do that work. And so Donna Berry, Sam Clark, John Crowley, and Michiel Scholes were kind enough to share the responsibility for doing this work. I'll confess that any of the mistakes shouldn't reflect on them. It probably goes to me. Vitelli did fund this work, so in the strict sense of the word, there's a conflict of interest between myself and the company. The only opportunity they had to influence the outcomes was through a review of a draft article, and that was provided to them to avoid any unintended disclosure of their proprietary intellectual property. So the technology basically is to have a scale co-joined with a watering device or feed trough. The technology itself's been around for a long time. Brad Knapp and Pat Curry back in the 1980s positioned a walk-through scale next to a water point in a pasture to measure body weight on a daily basis of cattle, grazing cattle. It provides for the passive capture of body weight, or in the case of most of the devices used today, partial body weight. And there currently are several companies that market such products, but there's really been very little, if any, evaluation of the protocols that are used to process these data. And so that's the space that we came into, was really to look at those technology, to look at the value of those partial body weights. The motivation, certainly if we have more frequent weight records, it might increase the accuracy of the measurement of average daily gain and some predicted weights. And this gives us the opportunity then to shorten the period of time that animals would necessarily spend on performance tests. It's been proposed that management could make use of these data in a variety of ways. From the perspective of animal welfare, certainly if the collection is entirely passive, that imposes less stress on the animals and obviates a need for labor and reduces the amount of human-animal interaction. I think there's also some tremendous opportunities for experimental applications in studying various effects on growth patterns of growth. So the objectives of this study were firstly to determine the utility of partial body weights in predicting full body weight and average daily gain, and then to determine the length of a test period that was required to accurately measure average daily gain when using partial body weights. And this is another image of another technology besides the Vitale technology. So the data that we received from Vitale were partial body weight records, their prediction, and there was one partial body weight for each animal for every day the animal was on test. That's a summarization, if you will, of the hundreds to thousands of actual measurements of partial body weight that were captured through the system. From that partial body weight, that daily partial body weight, they predict a full body weight. And the data that we had recorded body weights taken every couple of weeks throughout the period of time the animals were on test. I should point out that all of these measurements are measured with some error, including the recorded body weights. Almost 9,000 animals evaluated over 63 or more day test period. These animals were in 35 different contemporary groups, spaced out around the world actually, with many different breed types. And in our evaluation, we focused on body weight at the beginning and end of the performance test period and on average daily gain over the performance test period. Should point out that the reason for this focus for the beginning and end of the test period was because those are the most influential observations in determining average daily gain. So I'm going to skip now directly to the results. With reference to the partial body weights, the interaction of contemporary group with the regression of recorded body weight on partial body weight was highly significant. That means that this transformation of partial body weight to recorded body weight depended on the contemporary group. Those regression coefficients spanned quite a large range with an average of 0.18 and a fair sampling variance, but they did explain roughly 90% of the variation. The partial body weights explained roughly 90% of the variation in the recorded body weight across the data. For 8 of the contemporary groups, the intercept of the regression equation was positive. And this, our interpretation is that this indicates that a simple multiplicative adjustment of partial body weight to transform that value to a full body weight would be biased in those 8 contemporary groups. If we shift the focus a little bit to the predicted body weights rather than the partial body weights, that interaction with the regression on predicted body weight was again highly significant. The p-value was just slightly larger than the expected value of 1 and not statistically significantly larger. And the predicted body weights had a slightly higher correlation with the scale weights than did the partial body weights. Deviations of predicted body weight at the beginning of the test from the measured on-test body weight were significant in about half the contemporary groups and ranged from nearly 30 kilos to minus roughly 10 kilos. So a 40 kilogram range in terms of the deviations between the scale weight and the predicted full body weight. Should point out that we can't tell you which of those was actually responsible for the deviation. Going to shift gears to the end of test. And again, there was an interaction with contemporary group in terms of this regression. And you can see that the regression coefficients range had quite a large range. Really not a lot different than what you saw at the beginning of the test. And again, a multiplicative adjustment to predict recorded body weight from partial body weight would produce biased estimates of the recorded body weight. For the predicted full body weight, as we saw previously, the interaction with contemporary groups was highly significant. The deviations were just slightly smaller than they were previously. So I've John Crowley developed this visual image of the correlations between the partial body weight and the scale weight, or the measured recorded body weight, and the predicted body weight and that weight that was taken in a chute in the conventional way. And through the color coding says the values highlighted in yellow are pretty good. And as it moves down the color scale toward purple, those values get worse. And you do see some improvement in the prediction as you go from the beginning of the test, or from partial body weight to the predicted body weight. In both cases at the end of the test. These correlations are all very, very high, probably in almost many cases not different than 1. And at the end of the test, for some reason, there seems to be a little more variation in the correlation between the partial body weight and the actual body weight at the end of the test. The predicted body weight seems to clean up many of the problems that are present in the partial body weights. Again, the tendency for that regression on partial body weight, there was a tendency for that regression on partial body weight to increase over the test period, suggesting that even within a contemporary group, a single constant adjustment might not be entirely sufficient to convert partial or predicted full body weight to the chute weight. This slide shows the correlations from average daily gain, the correlations of partial body weight and predicted body weight averaged 0.81 and 0.78. So the correlation really aren't different. They're 0.8. And you can argue that that's whether or not that's really indicative of them being a different trait or not. Average daily gain from predicted body weight was generally higher than the average daily gain from recorded body weight. Although in most cases, as you can see from these little bars, that increase may not have been very great in a lot of cases. So again, here, much the same sort of graph, but now for average daily gain. The first column being the predicted weight, average daily gain calculated from the predicted full body weight and the reported body weight. The second column, the predicted weight and the partial body weight. And the third column, the recorded body weight and the partial body weight. And pretty strong agreement internally with the data captured by the system. The prediction algorithm works the way you think it does. Less strong agreement with the recorded chute weights. So to determine the length of test, we made use of the predicted full body weight. We thought that value was perhaps minimally better than the partial body weight itself. I'd point out that we did things a little bit differently than has been done in the past. Previous studies used the part-whole relationship between a shorter test period and the full test period to calculate how long the test period should be. That part-whole relationship has an asymptotic correlation of 1 as you approach the length of the full test period. And so we'll always provide something shorter than the full test period as being an appropriate or adequate length of test. We suggested that you use model order dependent regression. So you break the test period into 2 parts and regress those 2 parts on the full test period using model order dependent sums of squares, so that the latter part of the test period was adjusted for the first part, but the first part was not adjusted for the latter part. Using this strategy, we found that 35 and 43-day test periods were not adequate to measure average daily gain, but that a 50-day test period seemed to work sufficiently well. It explained about 80% of the within contemporary group variance And perhaps more insightfully, the remainder of the test explained only about 2% more. It'd be remiss to not point out that non-statistical factors also affect the decision regarding the length of test period. So in conclusion, when the capacity of your test facility is limited, or limits the number of animals that can be tested, a 50-day test period and testing more animals is likely to be a better procedure in terms of statistical power for any particular hypothesis than reducing the number of animals that are tested. If the number of animals that are to be tested is the limiting factor, then we'd recommend that you test them for a longer period than 50 days. We'd note that the adjustment from partial body weight to full predicted body weight likely changes over the test period, and that this prediction of full body weight over a test period is likely to have acceptable accuracy for many applications. I have a few comments besides, and these are sort of my personal opinions as I work through this process. In a research context, I think researchers need to control their own data. To me, I get nervous when we have black boxes that edit our data and predict or do predicted values. And in these data, the partial body weights, as I commented before, are summarized to a daily value, and I have no idea how that summarization was done. So you have a number of observations for an animal that range from a few hundred to a few thousand each day. Thus, the means of, if it's a mean for those animals, those means would vary in accuracy, and you might want to take that into account. I think when you do that summarization, you lose opportunities for research, particularly if those values are stamped with a timestamp that would then allow you to associate a certain animal behavior, whether it's eating or drinking, with other temporal values such as the temperature, precipitation, so on. Mathematically, I think you could use the partial body weights to predict a residual feed intake or residual feed intake and gain phenotype without much difficulty and without the necessity of doing any sort of transformation of that phenotype as it was collected. I think we probably need to do some more research to be able to put these partial body weight observations in the context of genetic evaluation. I'm not fully convinced what all of that research needs to look like. And finally, as I listened to Scott Spidel's presentation the other day, or 2 weeks ago, I was curious about whether or not the partial body weight information would be efficacious in monitoring for the onset of the bovine congestive heart failure syndrome that he spoke about. If those animals that are experiencing this congestive heart failure show decreased performance or decreased visits to a water or feed facility, then you might see negative residuals show up with increasing frequency toward as that heart failure became more severe. But I have no idea if that's true or not. And I guess I'd take any questions. And
All right. Thank you very much, Mike. If anybody has any questions, you need to put them in the Q&A section if you would, and we can entertain those. So just type away at your questions. So, Mike, I was curious on the in terms of the contemporary groups. If I remember right, the contemporary groups you said were not just in the U.S. Some of those were outside, and even regardless. So, how much variation was there in kind of the management practices in those contemporary groups in terms of types of feeds and things like that? Was that vastly different? Do you have any idea if that may have played a role?
we did not have that information, Derek. I mean, they clearly varied in the breeds that were in those contemporary groups. And there was everything from breeds that we would expect to be fairly high performing to breeds that we would expect to have in North American standards fairly low performance. I'm sure from personal experience, I'm sure that the rations vary considerably in their energy density. But I don't have any solid insight as to why those contemporary groups would be as different as they are, or as not different as they are.
Okay. And just in general, based on your conclusions at the end, you're not suggesting we're to the point now of being able to just submit those predicted weights into genetic evaluations, just to clarify that, correct?
Well, the predicted weight, because it's a prediction, would have a lower variance than the shoot weights. So is it enough lower that it matters? I don't know. There may be other reasons why you wouldn't do that. But I'd like to see a demonstration of those weights going into a prototype genetic evaluation system before I just woofed them into the Angus evaluation or the IGS evaluation or any other evaluation and just applied them like they were fine.
Yep. All right. Any other questions? Well, I'm going to give you a chance. Okay. Let's see what we've got. All right. So, okay, this is Juan.
I got a question from Juan.
Yep.
And it has to do with how lameness might affect the data. And I have no idea, Juan. None whatsoever. That was not part of the data set. It certainly would be interesting to know. I think there's a tremendous amount of opportunity with these data. And I just don't think that we as an animal science research community have explored those opportunities very well. And some of that is because we don't get all the data. The companies marketing the technologies do some data reduction before we ever see it. And for the bull test station operator, that might be appropriate. They may not have the expertise to do that data reduction themselves, and they probably don't care. But as a scientist, we care. And so I'm thinking I'd like to see all the data. I don't even know today if some sort of trimmed mean is better than the median.
Yep. All right, guys, in the interest of time, I'm not seeing any more questions come through, but so if you have any questions, you can certainly email any of us in terms of Bob Weber, Matt Spangler, or myself, and we'll get them to the appropriate places. So would welcome those. Remind everybody, our last session is next week, October 26th, and Matt's going to be hosting that one. Going to hear from Andrew Cromie, who's going to talk about some work we've been doing with BIF and some of the different international groups, ICAR in particular, on building an international wiki on data collection and genetic evaluations. And then I'm going to give a few thoughts on why should we really care about what the rest of the world is doing, and they're doing some pretty innovative things, so want to just discuss a few of those. And then we're going to also get our official invitation for the 2023 Beef Improvement Federation Symposium. It's going to be up north this year on July 3rd through the 6th, and if you are guessing or wondering, yes, that's tied to the Stampede, and so we'll hear all about that next week as well. So I would like to thank Mike and Juan again. We really appreciate you fellows joining us today and giving us an update on some interesting things happening these days in the beef cattle genetics world. So thanks a bunch,
Thank you.
and we'll see everybody next week.